Usual, begin recording and go to screen sharing. And we're going to talk about chemistry some more today. We're almost done, but not quite. So should plan on having our test on chemistry next Monday. Tomorrow at the beginning of class, like we did last time, we'll have us go over a short study guide about the main topics and a couple of sample questions related to chemistry. So we have one major category of what are sometimes called biomolecules left. And biomolecules are organic chemicals that are really only found in living things. And so we've talked about carbohydrates, we've talked about proteins, we've talked about lipids. And these are all things that are found in very high quantities in living things. In fact, they're found in such high quantities. These are the things that you see when you look at a nutrition label. Most of the calories that you get from eating the various foods that you eat come from the carbohydrates, the proteins, and the lipids. Even though we're going to talk about DNA today, compounds that are made from nucleotides, and we're going to talk about vitamins, and these things are important, they don't really contribute a lot of calories to your diet. If we took all the DNA out of the food that you're eating, and if we took all the vitamins out of the food that you're eating, there would be some consequences, but you wouldn't lose weight. They don't contribute a lot of calories to what you're eating. That's the carbs, the proteins, and the lipids. So let's begin by looking at what we're going to call compounds made from nucleotides. Now, if you're looking at our book or a lot of books, this category is just called nucleic acids. We're not going to call it nucleic acids because some of the things that are made from nucleotides are nucleic acids, but some of them aren't. So that name is a little bit misleading. So for that reason, we're going to call this compounds made from nucleotides. Now, keep in mind when we think of a nucleotide, we want to think of a protein, I should say an amino acid or a monosaccharide. So a nucleotide is a small unit. We're going to use it to build something much larger. So if we look at nucleotides, each nucleotide has 3 different parts to it. It has a monosaccharide that has 5 carbons in it. And this monosaccharide can be either deoxyribose or ribose. Depends which molecule we're looking at, which nucleotide. It has a phosphate group. So remember, a phosphate group is that PO4. And that PO4 sometimes has some hydrogens and usually has a negative charge. The phosphate group is pretty much always the same. And it has what's called a nitrogenous base. Nitrogenous means it has nitrogen in it. And this nitrogenous base either has the amino group, that NH2, or a very similar group called the amine group that's just NH. And the reason that this is called a base is because when we have this NH2 or this NH, this is slightly basic. So later today, we're going to talk about water and we'll talk about acids and bases. So you've probably heard of something called alkaline batteries. Alkaline batteries are basic. So here's our 3 parts of a nucleotide. This kind of light tannish, this is our 5-carbon monosaccharide. Since it has 5 carbons, it's sometimes called a pentose sugar. Penta refers to 5. We have a nitrogenous base. In this case, it's adenine. As we'll see in a minute, there's actually 5 different possibilities for the nitrogenous base. And it has a phosphate group. And notice the phosphate group is shown with an O, with a negative charge. And it's actually shown with 2 O's with negative charges. So this phosphate group is ionic. And this is very important because this means if you put some DNA into an electric field, something where there's an electric current running through the region, the DNA separates into different pieces. And it separates basically according to its size. So I'm sure you've probably all seen some program where they're showing you a sheet with all these little bars on it, and they're telling you that this is the DNA profile of the suspect or maybe the criminal. So this is how we get that type of pattern. And we'll talk about this in more detail towards the end of the semester when we look at DNA. But basically, we run an electric current through some DNA. The DNA separates into pieces that are different sizes. And if we look at your DNA and compare it to my DNA, or even your sister's DNA or your brother's DNA, your DNA won't be exactly the same as any other person. The pieces that we get are a little bit different size in me than they are in you. And they're a little bit different size in me than they are in any of my 3 brothers. So unless you have an identical twin, if we do this with all of your DNA, we can tell you apart from any other person in the world. Now, if we only do a little bit of your DNA, if we just take a little chunk of it, think of it like looking at a physical characteristic. If I look at your complete physical characteristics, we could identify you as you. But if all I look at is your eye color and your hair length, there's probably a lot of people around the world who have your eye color and your hair length. So if we only look at part of the DNA, we may not get a conclusive identification. But we can only look at DNA at all because these phosphate groups have charges on them. So when we look at the nitrogenous bases, there are 5 different nitrogenous bases, and we hardly ever write them out. Adenine is almost always abbreviated with an A, guanine with a G, cytosine with a C, thymine with a T, uracil with a U. Notice these are always capital letters, they're never lowercase. So let's look first at nucleic acids, since this category is sometimes referred to as nucleic acids. And as I said, we're not going to call it that. So here's a small piece of a nucleic acid. And the reason I say this is a small piece is this is a section of DNA. And here our sugars are shown, our 5-carbon monosaccharides are shown in blue. Our phosphate is kind of the peach colored, and then they use different colors for the 4 different bases. But if we look and we count, we have 4 phosphate groups, we have 4 different bases, we have 4 monosaccharides. So this is 4 nucleotides. Now, the reason I say this is a small molecule, lots and lots of atoms here, but if you look at the human body, the average DNA molecule is about 150 million, not just nucleotides, but nucleotide pairs long. So this is really just a small section, and it's not showing the pairs. There would be another strand next to this. So when we look at DNA, notice that if we start down at the bottom, here's our 5-carbon monosaccharide. It's bonded to a phosphate. That's bonded to the next 5-carbon monosaccharide. That's bonded to its phosphate. And we keep on going up the line. We go from a monosaccharide to a phosphate, from a monosaccharide to a phosphate. Now, the phosphate group is attached to the 5th carbon of this 5-carbon sugar. So the part of this chain that ends with the phosphate is called the 5' end. The nucleotide is attached to the 1st carbon, And the third carbon is this oxygen down here on the bottom that would attach to the next phosphate group if there is one. So this end that ends with the sugar is called the 3' end. So we have the 5' end on one end of our DNA chain, the end that ends with the phosphate. We have our 3' end on the other end. This is the end that ends with the monosaccharide. Now, when we look at this, when we think of the order of our DNA, we always start at the 3' end and think about what would be next to this. So this is a very small piece of DNA. Notice they have, for their nucleotides, their nitrogenous bases, they have one adenine, one guanine, one cytosine, one thymine. In real life, you don't necessarily have these nitrogenous bases in equal numbers. In some parts of the DNA, you might have more thymines. In other parts, you might have more guanines. But when we analyze human DNA, if we would analyze all the A's, C's, G's, and T's in your DNA, we wouldn't come out with 25% of each. We don't have equal amounts. And there's no other living thing that does, as far as I'm aware, either. So when we think of a nucleic acid, it's a big molecule. If it's DNA, as I say, the average DNA molecule is 150 million nucleotides long. There are big DNA molecules in human cells that are more like 300 million nucleotides long. There are also small ones that are only 20 million or so. So they range in size, but they're very big molecules. So we're going to see this pattern repeated over and over again. Phosphate to sugar, phosphate to sugar, phosphate to sugar, phosphate to sugar, and then the nucleotides stick out. Sometimes this is compared to kind of half of a ladder. If you imagine that the sugars and the phosphates are the one side of the ladder, and then the bases that stick out are kind of half of a step. They're only half of a step because, remember, I said this is actually connected to another molecule, which would be up above this. Now, if we look at RNA, RNA is very similar, except RNA has ribose in here for the little blue monosaccharide. This is actually deoxyribose, since this is DNA. And RNA doesn't have thymine. RNA has uracil. So DNA and RNA are almost exactly the same. So if we look at DNA and we think about those nucleotides, we always have deoxyribose sugar. The D stands for deoxyribo. So that tells us the type of sugar that we have here. We always have a phosphate group. And then we have one of the nitrogenous bases, either AC, G, or T.
NH3.
So when we look back here, notice that each nucleotide has deoxyribose sugar, a phosphate group, and then it has one of the nucleotides. So a phosphate group again, deoxyribose sugar, one of the nucleotides. Phosphate group, deoxyribose sugar, one of the nucleotides. So that's the common pattern that we see. The sugar is always deoxyribose sugar. The phosphate group's always the same. It's only the base that's different. Could be A, could be C, could be G, could be T. And really, when we look at one of those DNA profiles, like I talked about before, the difference between DNA profiles is really the order of the A's and the C's and the G's and the T's. My order of A, C, G, and T is not exactly the same as your order of A, C, G, and T. And that's what we're telling apart when we look at the DNA. Now notice, when we think about the DNA really always pretty much does the same thing. It's a set of instructions, but the instructions are written in a code. They're not They're not super straightforward, but later we'll see how to read that code. Fortunately for us, other biologists have already figured out. And these instructions only tell the cell one thing. These instructions tell the cell the primary structure of the proteins. Now remember that the primary structure of proteins is just the order of the amino acids. So, throughout the last few days, I've said one way to think about proteins is they're kind of like words. The amino acids are like letters. There's 20 amino acids, not 26, but they're kind of like letters. We can use them to spell proteins. So some people say that the DNA is really very similar to a dictionary. Your DNA is the dictionary of all the proteins that your cells can make. And it tells the cell how to spell those proteins, what the order of the amino acids is. If we look at ribonucleic acid, or RNA for short, it has 3 different parts. It always has ribose sugar, so not deoxyribose. It always has a phosphate group. That's actually the same as in DNA. But where DNA has ACG or T, RNA has ACG or U. It never has T, thymine. And RNA, the main thing that we're going to talk about is that RNA basically translates the instructions from the DNA and helps the cell to build proteins. So you really have to have RNA and DNA for the cell to work. And in fact, there are some antibiotics like tetracycline, and tetracycline works by interfering with the RNA in a bacteria cell. So the RNA can't make proteins. And without proteins, cells can't do anything. Now, the reason that I don't like to call this category of chemicals nucleic acids is because everything that's made from nucleotides isn't a nucleic acid. Another type of molecule that's made from a nucleotide is something called adenine diphosphate or adenine triphosphate or cyclic adenosine monophosphate. So if we look at adenosine, all of these have adenosine in them. Adenosine is just adenine, so that's that A from up here. And it's adenine combined with ribose. So notice ATP is made from a RNA nucleotide. And a nucleotide, we said, has ribose sugar. So this red is our ribose sugar. It has a phosphate group, this yellowish, and it has a nitrogenous base, either ACG or U. In the case of ATP, it's always A, it's always adenine. But here's how ATP is built from a nucleotide. If we think about DNA and RNA, DNA is built by attaching one nucleotide to another nucleotide, and then attaching the second nucleotide to the third, and the third to the fourth, and the fourth to the fifth, and all the way down until we have millions of them. If we look here, what we've done, instead of adding more nucleotides, we've just added more phosphate groups. So ATP is adenosine triphosphate. So adenosine is the adenine and the ribose. Triphosphate tells us we have 3 phosphate groups. Now, ATP is very important in cells because ATP is the molecule that cells use to get their energy. Sometimes people think of glucose as being how cells get their energy, but when your cells use glucose, they actually use glucose as energy to build ATP. And then when your cells want to do anything, if they want to move your muscles to, move your hand to get you up out of your chair to walk across the room, your muscles have to use ATP. So ATP is your direct source of energy. this is what you use. If your cells run out of ATP, since this is what they use for energy, your cells start dying. This is one of the things that happens during a heart attack typically, is that muscle cells in the heart don't get enough oxygen. We'll see later that you need oxygen to make ATP, and without enough oxygen, heart muscle cells start to die. Now, the way that we get energy from ATP, if we don't show quite as much detail, so here's our adenine over here, this green ring structure. Notice that here it was blue, but it's a 6-member ring connected to a 5-member ring. So we can see 6 members here, 5 members here. Remember, the points of the lines represent atoms. Down here we have our 5-carbon sugar, ribose, and we have 3 phosphate groups attached. Now there's a lot of energy in the chemical bond that connects the 3rd phosphate group to the 2nd phosphate group. And what we're going to do is what we always do to take a big molecule and break it down into smaller molecules. We're going to do something called a hydrolysis reaction that we talked about the other day. So we're going to add some water, H2O. Notice that water and ATP are both at the beginning of the arrow, so they're both reactants. So we're going to add some water and we're going to stick an OH back onto one of the phosphate groups, and we're going to stick an H onto the other one. And when we do that, now we have a loose phosphate group. Since this phosphate group doesn't have any carbon with it, it's sometimes called an inorganic phosphate, or as they have designated up here, a P with a lowercase I behind it. And what we have left of our main molecule is now called ADP, adenosine diphosphate. So we still have our adenine, we still have our ribose, but now we only have 2 phosphate groups. Di means 2. So if we do this, we get some energy. When we break this chemical bond that connects the 3rd phosphate group to the 2nd, the energy in that bond is released. And now this energy can be used to do work. As I said, it can be used to move your muscles, but it can also be used to transport materials around your body. It's used when your brain cells do something that we call thinking or making memories. That takes ATP energy. In fact, actually, even though your brain is relatively small compared to a lot of parts of your body, your brain uses a lot of the ATP. Now, the problem is, as soon as you use ATP, what do you have left? ADP and a phosphate. Now, you could eliminate these in your waste, but instead, what living organisms do is they save these. And then they rebuild their ATP. They use their leftover ADP, their leftover phosphate, and they stick them back together. But to do this, you need energy to rebuild that bond connecting the 3rd phosphate to the 2nd energy. So where do we get that energy? From glucose. And we're going to oxidize glucose. And if you oxidize something in chemistry, this means that you react it with oxygen. And this is the same reaction that happens when you burn something. So basically we burn glucose. So think about, burning a log in a fire. You get energy, you get light, that's a form of energy. You get heat, that's a form of energy. So in the case of glucose, when we burn glucose, when we oxidize it, we're going to get energy that we're going to use to stick ADP and phosphate back together. And when we stick them back together, we make ATP again. So instead of having to eat a lot of phosphorus in your food, you basically just recycle the phosphate groups and the ADP and the ATP. You break the ATP down. After you break it down, you put it back together. After you put it back together, you break it down again. So this cycle happens continuously. And there's another molecule abbreviated NADP. And NADP, the DP at the end stands for diphosphate. So this is also something that has extra phosphate groups that is built from a nucleotide. Now let's think about vitamins very briefly. Vitamins are organic molecules. They do a lot of different things in the body. Back when you were in grade school, you might have learned the vitamins in your science class, and you learn what they do. But it turns out that what we learn in grade school is really just kind of touching the surface. Usually when we first learn about the vitamins, what we learn about is their main function, maybe 2 or 3 main functions. But it turns out lots of vitamins have many, many functions. And not only that, when we look at the vitamins, most vitamins come in more than one form. If we look at vitamin E is actually 8 different chemical compounds. Now they're all very similar to each other, but some of them are more efficient than others. Some of them work in certain parts of a cell and others work in different parts of the cell. Vitamin D has several different chemical forms. So most of these chemicals exist in several different versions or varieties. And vitamins are usually coenzymes. Now remember, we've talked about enzymes a little bit in the last day or two, and we said that enzymes control chemical reactions. They speed them up. Most enzymes need certain coenzymes to work properly. Most enzymes in your body only work with one metabolic reaction. Most coenzymes work with dozens, or in a few cases, hundreds of metabolic reactions. So every enzyme works in a different reaction. Every enzyme does something a little bit different, but coenzymes work in lots of reactions. They do the same thing, the exact same thing in every reaction. So some coenzymes include some things like NAD and NADP. And the D here is dinucleotide. And the N comes from the chemical name for one of the vitamins, a vitamin called niacin. There's another coenzyme called FAD, and this is built from the vitamin riboflavin. So the F comes from the flavin. And usually when they're coenzymes, what vitamins do is they pick up an electron or sometimes an entire functional group from one molecule, and they transfer that electron or that functional group to a different molecule. Now, when we look at the vitamins, there are a lot of different vitamins, but they fall into 2 basic categories. They're either fat soluble or they're water soluble. Fat soluble vitamins are hydrophobic. They don't mix very well with water because they're basically lipids. Now notice you have to be careful if you're taking vitamin supplements, because if you take a supplement of a fat soluble vitamin, you can actually overdose on some of these fat soluble vitamins, because if you consume a large quantity of them, they get stored in the fat in your body. And for instance, vitamin A is actually toxic in very large quantities. Usually it leads to mental problems first, but eventually it can kill you if you eat too much of it. On the other hand, water-soluble vitamins are hydrophilic. They have mostly polar and ionic bonds. Now, the water-soluble vitamins are pretty easy to keep track of because there's really only 2 vitamins, well, 1 vitamin in one group that are water soluble. Vitamin C is water soluble. And the B vitamins, and there's a bunch of B vitamins, 8, I believe. The B vitamins are water soluble. So when we look at the water soluble vitamins, if you take a tablet, a vitamin, and it has a lot of vitamin C in it, then you don't get poisoned by that. Why? Because the vitamin C is water soluble. So any excess that you don't really need right now, your body can store a small amount, but just a small amount. But if you take a lot of vitamin C, it'll just be eliminated in your urine. And in fact, if you look at your urine, urine should be pretty much colorless. If your urine has color, then typically it means you might be taking too much of some vitamins, or you might be eating too much protein, because if you eat a lot of protein, you have to eliminate the amine group. That's a waste product most of the time, and that can make your urine colored. So if your urine is colored, you might be taking too much of some vitamins, you might be eating too much protein, or you just might be dehydrated. Those are probably the most common dietary reasons why your urine would be colored. I used to work at a health club a little while, and we said that the weightlifters had the prettiest pee because they were all taking amino acid supplements. And if you take a lot of amino acid supplements like they did, most of the amino acids just come out in your urine and make it nice bright yellow or orange. So vitamins are crucial, but the vitamins are kind of like the ATP that we mentioned a moment ago. We said ATP gets broken down, then you rebuild it. It gets broken down, then you rebuild it. So you don't really need a lot of phosphorus in your diet. The vitamins don't even get broken down. When they work as coenzymes, when they pick up an electron and they transfer it from one molecule to another, they're not damaged or changed. So when they pick up the electron, they're a little bit different, then they drop it off, they're back to the way they were, and they can head out someplace different in the cell and pick up another electron. And they can do this over and over and over again. Now, since molecules are physical entities, vitamins and enzymes do wear out eventually, kind of like the tires on your car have to be replaced every once in a while. But vitamins and enzymes can last a long time. Now, the last two things that we want to talk about that are important chemicals that are found in living things are both inorganic. They're not necessarily bonded to carbon. So one of these is minerals. So minerals include sodium, potassium, magnesium, calcium, iron, copper, zinc. Some plants have silicon in them. And minerals usually do something similar to coenzymes. When we say that vitamins are coenzymes, coenzymes are usually organic molecules. I say usually because chemists can't quite agree on how they're going to name these things. Minerals are similar to coenzymes, except they're usually called cofactors. And just like a coenzyme, co means with, a coenzyme helps an enzyme do its job, helps it speed up the reaction that it's involved with. Cofactors do the same thing. So there's one enzyme called DNA polymerase. And notice enzymes almost always end in ASE, not always, but almost always. And this needs magnesium ions and zinc ions if it's going to work. If it doesn't have magnesium and zinc, DNA polymerase can't do its job. So when we talk about coenzymes or cofactors, the minerals that are helping out, what we want to keep in mind is that these things are really crucial for metabolism to work correctly. And this is why if you have deficiencies of these minerals or vitamins, certain metabolic reactions don't occur. So you get certain illnesses. If you don't get enough vitamin C is needed to, it's a coenzyme in some of the chemical reactions that make collagen. Well, remember, we talked about collagen a little bit the other day. Collagen is a protein, and there's so much of it in your body. We said if you took everything else out of your body, every other molecule, you would have a perfect outline of every organ and of your whole body. So if you can't make collagen, if you don't have enough citric acid for a long time, as collagen in your body starts to wear out, you can't replace it. Your body literally starts to fall apart. You start to lose chunks of your skin. On the inside of your body, parts of your blood vessels can break off. So you get what we call embolisms, or you could have a stroke, you could have a heart attack. So just because we don't need a lot doesn't mean these things aren't important. And keep in mind, when I say we don't need a lot, for most of these vitamins and minerals, we need milligrams a day. Now, to give you an idea of how much a milligram is, there are about 25 grams in an ounce. And a milligram is 1,000th of a gram. So that's, 0.0004 of an ounce. It's really not very much at all. But if you don't get that, you can get certain diseases. And if you go back to the early 1900s, these diseases were very common. If you don't get enough vitamin C, as I mentioned, usually the first disease that you get is a disease called scurvy. And one of the symptoms of scurvy, you have a lot of collagen in something called your periodontal ligaments. And your periodontal ligaments are what hold your teeth in place. If you ever started to lose a tooth when you were a little kid, those ligaments loosen when you get ready to lose a tooth. So sometimes you can wiggle your tooth, or sometimes your tooth is just hanging by a strand. That strand is those periodontal ligaments, but those things are mostly collagen. So when people get scurvy, one of the first things that's noticeable is their teeth fall out. If you don't get enough of some of the other vitamins and minerals, you can get things like beriberi and rickets and pellagra and diseases like that. Most of these diseases are pretty rare today, but if you would go back to the early 1900s and before, they were very common. That's how a lot of people died, was from basically dietary deficiencies. We didn't necessarily know it at the time. Now notice there is one other thing that minerals are commonly used for. A lot of living organisms use minerals to build parts of their body. For instance, if you look at our bones have a lot of calcium and phosphorus. And there are animals like oysters who make their shells out of calcium. Not really very much phosphorus in there, but a lot of calcium. So some living organisms use minerals for things besides cofactors. But notice there are a lot. We have bones, but if you look at something like, say, a sponge, which is a type of animal, or a jellyfish, they don't have very much calcium in their body. They have a little bit because calcium works as a coenzyme, a cofactor, but they don't have any bones or anything like that made from calcium. And then finally, we've saved till last the molecule that there's actually more of in your body than any other type of molecule, and this is water molecules. Now, remember, when we think about a water molecule, one of the things we said a few days ago was if we look at the bond between the oxygen atom and each of the hydrogen atoms, this is a polar covalent bond. And we said in polar covalent bonds, electrons aren't shared equally. In the case of oxygen and hydrogen, oxygen has a stronger attraction for electrons than hydrogen does. Remember that attraction for electrons is called electronegativity. That's a a measure of how much something attracts electrons. So this means when oxygen forms a bond with hydrogen, the oxygen has a delta minus charge. And remember, we said that's a partial negative charge. It's not a complete negative charge like we would see in an ion. The hydrogen atoms have a delta plus charge. And again, it's not a complete positive charge like we would see in a positive ion, but it's kind of a partial positive charge. Now, because they have this delta plus charge, hydrogen atoms are very strongly attracted to any electrons they can find. Remember, electrons have a negative charge. And when we were talking about protons and electrons and bonds, we said opposite charges attract. So hydrogens that have a partial positive charge are strongly attracted to electrons. And because of this, hydrogens actually make a different type of bond, one that we haven't talked about yet. The hydrogen atom connects to an oxygen atom, but the hydrogen atom in one water connects to the oxygen atom in a completely different water molecule. So here we have some water molecules. Notice this is one of those space filling diagrams like we talked about earlier. Oxygen is much bigger than hydrogen. And so the oxygen is connected to two hydrogens. The oxygen has a partial negative charge. Each of the hydrogens has a partial positive charge. So where's the nearest place where a hydrogen that has a partial positive charge can find a negative charge? Well, a neighboring oxygen. So what we see is these dotted lines. And these dotted lines are hydrogen bonds. Now notice this is very different from polar covalent bonds or polar non-covalent bonds that we've talked about before. Polar covalent bonds are the bonds between the oxygen and the hydrogen in this molecule. Non-polar covalent bonds are also bonds between two atoms in the same molecule. A hydrogen bond is a bond between a hydrogen in one molecule and an oxygen in a completely different molecule. So this is a type of bond between two different molecules. And what this means is when we look at water, these hydrogen bonds hold all the water molecules together. Now, hydrogen bonds are very weak. And because they're weak, they're very temporary. Because if we look at water molecules or any kind of molecules, not just water, but if we look at these water molecules, they're continually bouncing around, they're vibrating, they're moving. And so if we look at this water molecule down here in the lower right, when it moves around, its oxygen might not be next to this hydrogen anymore. So that means this hydrogen bond, it's broken. But this oxygen might move up so that it's next to this hydrogen. So a new hydrogen bond forms. So if we would look at a bunch of water and we could keep track of where all the hydrogen bonds were, the hydrogen bonds would constantly be shifting. They'd always be there, but first a hydrogen would be bonded to an oxygen in one molecule. Then if we looked a second later, it would be bonded to an oxygen in a different molecule. Now, water is very unusual. Hydrogen only makes hydrogen bonds either when it's bonded to oxygen or to nitrogen atoms. So most things don't make hydrogen bonds. So that means that most things don't have bonds between different molecules like water does. And holding these water molecules together, this gives water a lot of unique properties that nothing else has. And it makes these water molecules, it makes water as a chemical crucial for living organisms. We haven't discovered life on any other planets yet, but we think if there is alien life, it would have to be based on water. We're not positive about that, but from what we can tell, water is the only molecule that would work like this. So when we look at this, these hydrogen bonds are really crucial for water These hydrogen bonds give water properties that no other common element has, no other common compound, I guess I should say, since element has a specific meaning in chemistry. So one of the properties that water has is water is sometimes called the universal solvent. A solvent is something that will dissolve other things. And universal means that water can dissolve anything. Well, that's an exaggeration. Water doesn't dissolve everything. But here's what we do know. Water will dissolve almost all ionic substances and almost all polar substances. The only exception is if this ionic substance or polar substance is really a huge molecule. Think about water. Water only has 3 atoms in it, H2O. So if a molecule gets really big, water may not be able to dissolve this. A good example of this is thinking about carbohydrates. Think about starch. Starch is somewhere between 30 and 300 glucose molecules bonded together. Now, glucose is polar. Glucose will dissolve in water, but there's a problem. It's so big, it doesn't dissolve in water very well. If you've ever made gravy and you've tried to thicken the gravy, one way that you can thicken gravy is you can put in some starch. And when you put in the starch, what do? You have to stir it up, first of all, so it doesn't form lumps, but also to get it to dissolve, because if you don't stir it up, the starch just sits there. So you have to stir it up, you have to heat the water, and if you do this, you can get some starch to dissolve and you can thicken your gravy that you thought was too thin. But if you put starch in cold water, it doesn't really dissolve very well. If you don't stir it up, it doesn't really dissolve very well. So water can dissolve things if they're polar or ionic, as long as they aren't too big. If they get to be too big, then they don't dissolve very well. Now, when we talk about starch and gravy, we're talking about cooking, but the same thing is true in cells. Really large molecules, even though they might have lots of polar bonds or lots of ionic bonds, don't dissolve in water very well. Now, the other thing is, when we think about water, since water is polar, it doesn't dissolve most non-polar substances. Except now we're going to change our perspective. Water can dissolve a few non-polar substances if they're very small molecules. Like what? Well, like oxygen. Fish in the water breathe oxygen just like we do. And the reason they can breathe oxygen is because oxygen in the air dissolves into the water. When the fish exhale, they release carbon dioxide, and the carbon dioxide dissolves into the water. So even though it's not completely accurate to say that water is the universal solvent, it dissolves a lot of substances. And since water is the main molecule that's found inside your cells, for other substances, if they dissolve in water, they're going to have a different type of chemistry than if they won't dissolve in water. Now, another crucial factor. If you start heating something up and you measure its temperature, when you measure the temperature, you're actually measuring how fast the molecules are moving. And so when something gets hotter, the molecules are moving faster. But remember what we said a few minutes ago. Most substances don't have hydrogen bonds, so the different molecules aren't connected to each other. So in water, to kind of separate the molecules and to get them moving faster, it takes a lot of heat. Think about what happens if you start heating water in a pan. You're going to make noodles. If you turn on the heat, the pan gets hot really fast. But for a while the pan is super hot, the water is still kind of lukewarm. You can stick your hands in it if you want. And the reverse is true when you get done, cooking your noodles. If you pour the water from your pan out into a different container, the metal pan will cool down very quickly, but the water stays hot a long time. Well, why is this? It's because it takes a lot of energy to change the temperature of water. If we're going to raise the temperature of water, the water has to absorb a lot of energy. If we're going to lower the temperature of water, if we're going to cool it down, we have to remove a lot of energy. So this is crucial because if we look at the Earth as an environment for life, about 79% of the surface of the Earth is covered by water, mostly the oceans. So what does this mean? This means that the temperature of the Earth is relatively stable. The temperature of the Earth doesn't go up and down a little bit. You know, about the hottest it's ever been on the Earth is 120 degrees Fahrenheit, a little bit warmer than that. About the coldest it's ever been is minus 120 degrees. So there's a difference of about 250 degrees Fahrenheit. Some planets like Mars, the temperature typically changes on Mars more than 100 degrees Fahrenheit every day. Why? Because there's no water. Now this can even be seen when you look at local environments. If you look at Chicago, you've probably noticed if you've ever seen a weather report, but you know, now that we're in the middle of summer and they tell you the temperatures, it's usually cooler down by the lakefront. Why is it cooler by the lakefront? Because it takes a lot of energy to raise the temperature of all that water in Lake Michigan. You know, in another month or two, when the air temperature will be 95 degrees or 100 degrees Fahrenheit, Lake Michigan will be maybe 70 degrees, 75 degrees Fahrenheit. Now, why is this? It's exposed to the same amount of sunlight as we are, but it takes a lot more solar energy to raise the temperature of water than to raise the temperature of the gases in the air or ground. So, you know, if you think about something like asphalt, it heats up very quickly. Water, very slowly. Now, this has a big effect on climate. If we look at Chicago, our summers are cooler than they could be if we didn't have Lake Michigan right next to us, because Lake Michigan absorbs a lot of the heat energy. Notice the reverse is true in winter. We always complain about our winters in Chicago. I guess we're just whiners, but if you look at our winters, during the winter, Lake Michigan has to lose a lot of energy. So if you've never noticed, you know, wait till next December, January, and if you watch a weather report, it's warmer down by the lakefront. Why? Because Lake Michigan hasn't given up all the energy that it absorbed over the summer. When our temperature outside might be minus 20, Lake Michigan is a nice warm 35 or 40 degrees. So, you know, we might not consider that warm enough to go swimming, but it's a lot warmer than minus 20. So this means that this affects our climate. So like I say, people from Chicago, I can say this since I live here, we're whiners. We always complain about our hot summers and our cold winters. But if you want to see what hot summers or cold winters are like, think about going straight east or straight west from us. Where we are, west works best, but go west from us and live in Des Moines for a few years, or go even further west and live in Omaha for a few years. What do we have that Des Moines and Omaha don't have? We have Lake Michigan. So guess what? The summers in Des Moines and the summers in Omaha are hotter than our summers. And the winters in Des Moines and the winters in Omaha are colder than our winters. And this is one of the reasons why in other parts of the country, like San Francisco is by a warm ocean current, and they have very mild winters, despite the fact, if you check on a map and you see where San Francisco is, they're actually pretty far north. You would expect their winters to be warmer, except they're by a big body of water. So their temperatures tend to kind of even out. Now, the other thing that this is related to, since if we look at our bodies and cells, our bodies and cells are also made of water. This means that our body temperature is relatively stable. It doesn't change a lot. Sometimes we think of our body temperature as never changing, but obviously it goes up a little bit when you have a fever. And what you may not be aware of is when you go to sleep at night, most people, their body temperature goes down a degree or two. Not a lot, but just like in winter, some people turn down the thermostat when they go to bed to save energy. When you go to bed, your body, in a sense, turns down its thermostat, saves a little bit of energy because you're not going to notice when you're asleep anyway. But sometimes in the summer, you wake up, and as soon as you wake up, you start to feel chilly. Why do you feel chilly? Well, if your body temperature went down a degree or two overnight, you were fine with that, you were asleep. But as soon as you wake up, you're thinking your body temperature should be 98.6. And since it's, say, 97.6 degrees, you feel chilly. Finally, there's a third property of water that we're going to focus on. And this might seem trivial because actually it only happens about once in every 10 million water molecules. So not very often at all. But in about 1 out of every 10 million water molecules, the water molecule ionizes. And the oxygen actually pulls the electron away from the hydrogen. Remember, I said the oxygen had a stronger electronegativity. So we get an OH with a negative charge. The negative charge is really by the oxygen. We get a hydrogen with a positive charge. So these are our two ions. And we can measure the concentration of hydrogen ions on something called the pH scale. Now, if we would look at the pH scale, pure water is considered to be neutral. And it's neutral because when pure water ionizes, we always get a hydrogen ion and we always get an OH ion. And so if we have equal amounts of hydrogen and OH, then this is a pH of 7. But there are some chemicals that are called acids. For instance, a few minutes ago, we were talking about deoxyribonucleic acid. Acids are things that make more hydrogens than OHs. And for chemicals that make more hydrogens than OHs, they have a pH lower than 7. So 6.5, 4.3, 2.1, it depends how strong they are. If we look at the stomach acids that help you digest your food, they have a pH somewhere between 1 and 2. Now, one of the things we have to be careful about when we're looking at the pH scale is it's similar to the scale that we measure earthquakes on or hurricanes. If we look at an earthquake that is magnitude 7 and we compare it to an earthquake that's magnitude 6, might seem like, okay, 7 versus 6, no big deal. Except when you go from 6 to 7, an earthquake is about 10 times as strong. So if we're thinking about going from pH 7, pure water, to pH 2, stomach acid, when we go from 7 to 6, there's 10 times more hydrogens. When we go from 6 to 5, there's 10 times more hydrogens again. 5 to 4, 4 to 3, 3 to 2. So this means when you go from pH 7 to pH 2, there's 100,000 times as many hydrogen atoms. So it's a much, much stronger acid. And in fact, the acid in your stomach is so strong, your stomach is protected by a thick mucus so that this acid doesn't damage your stomach. But if this acid gets out of your stomach, it can cause problems. So there are some people who have a disease called GERD. GERD is G-E-R-D, and it stands for gastro, which is the stomach, esophageal reflux disease. And in GERD, the juices in your stomach back up into your esophagus. The esophagus is the tube that brings the food down to the stomach. And when those acids back up, the esophagus doesn't have any protective layer of mucus. So those acids can actually dissolve a hole in the esophagus if the disease isn't detected in time. And if they dissolve a hole in the esophagus, what's around the esophagus? Your lungs and your heart. So they can cause very serious health problems. They can cause death if they're not detected in time. Now, other parts of your body have different pHs. So acids make more hydrogens than they make OHs. Bases, or we sometimes call them alkaline chemicals, make more OH than hydrogen. And they have a pH higher than 7. And again, every time we go up 1 unit, when we go from pH 7 to pH 8, there's only 1/10 as many hydrogens. If we go from 8 to 9, there's 1/10 times another 10. So if we look at the pH scale, the pH scale essentially goes from 0 to 14. So pure water is in here in the middle at pH 7. So human urine is typically slightly acidic, somewhere around pH 5. The acidity varies. It depends partly on, what you've been eating, what type of waste products are found in your urine. Tomato juice is around pH 4. Gastric juices, the stomach acids, are pH 2 or slightly lower. And one way to think about how acidic pH 2 is, not only does pH 2, not only could those stomach acids dissolve your esophagus, people who have an illness called bulimia, where they tend to eat food and then throw up, the stomach acids, when they throw up, can actually dissolve the enamel on their teeth. Now, if we go in the other direction, blood has a pH of around 7.4. So again, pH 7.4 might seem like, not that big a deal, still pretty close to 7. But this is actually about twice as many OH ions and only about half as many hydrogen ions. On the other hand, if we look at soap has a pH somewhere around 10. The exact pH depends on the type of soap. Some soaps tell you that they're pH balanced, and they try to get a pH that's a little bit closer to the pH of skin. Bleach has a pH of around 12. Now, let's just think about the blood and the stomach acids for a moment. Very different pHs. So different parts of the body have different pHs. Now, in your stomach, if the pH changes a little bit, you may feel some indigestion. You may not be able to digest your food very well, so you may get diarrhea or you may get constipated. But if the pH in the blood is off, it's very serious. Normal human blood, we say that it's a pH 7.4, but it's between about 7.35 and 7.45. And it doesn't seem like much of a difference, but if your blood pH gets down to about 7.33 or 7.48, you're going to be hospitalized, you're going to be in intensive care, and depending on how quickly they caught it, you might survive. But when your blood pH changes, remember when we were talking about proteins the other day, when the pH changes, that can denature the proteins. And if we denature proteins, we change their shape, they don't work like they should. And one of the important proteins in the blood is that protein that we keep coming back to, hemoglobin. Hemoglobin picks up oxygen in your lungs. It carries oxygen to other cells and drops it off. So if the blood pH changes a lot, you may not pick up enough oxygen in your lungs, or you may not drop it off in your other cells. So you can literally be in a room like you're in now where there's plenty of air and you can be suffocating because you're not delivering oxygen to your cells. So from a practical point of view, your blood pH has to be very tightly regulated. Now, when we have pHs that have to be regulated very carefully, we have something called buffer systems. And buffer systems are sometimes single chemical compounds, or sometimes they're pairs of compounds. But the key is in either case, whether it's a single chemical or a pair of partner chemicals, they're chemicals that resist a change in your pH. So for instance, in people, we have something called the bicarbonate buffer system. We have something called the phosphate buffer system. And we have the protein buffer system. And these are all found in human blood. And they do a very good job of controlling your blood pH so that it's between 7.35 and 7.45. Even the hemoglobin that we've been talking about on several different occasions provides a little bit of a buffering effect. So water has more important properties than we've talked about. These are going to be the three that we'll focus on. Water's ability to dissolve a lot of different chemicals, water's high specific heat, the fact that you have to add a lot of heat energy or remove a lot of heat energy to change water's temperature, and water's ability to ionize. Now, if you want to visualize hydrogen bonds and how they work, one way to do this is to think about what happens if you wax a car, or if you wax, could be a kitchen counter. If you wax something and then you spill water on it, what happens? The water beads up into little droplets. And this is because of the hydrogen bonds. All the water molecules kind of cling to each other. On the other hand, if you spill water on a surface that's dirty, the hydrogen bonds get overpowered by bonds to all the dirt that's on that surface. So the water doesn't cling, it kind of spreads out. So are there any questions about either compounds made from nucleotides or water or vitamins or minerals that we discussed today? Okay, I'll let you go in a minute, but tomorrow we'll have a study guide for the chemistry, similar to the study guide that we had for chapter 1. So we'll go over a few practice questions. So I'll have that posted before class. So if you want to get started on them before class so that you can potentially see whether your answers are correct or not. And we'll have a short assignment related to organic chemistry and biochemistry. And similar to last time, that assignment will actually be due Monday night. But if you finish it by noon on Sunday, I will grade it. And if you attach a note to it up by where you write your name and tell me you want to see it back, and I'll send it back to you so you can use that to study for the test. Are there any questions before we go?
So you said the test is Monday, right?
Yeah, the test will be Monday. I'll give you some more details tomorrow, but usually the chemistry test takes a little bit longer. So you'll probably have like an hour and 45 minutes. And like this last time, it'll probably be up 10 or 15 minutes early.
All right.
So I would recommend for this one, you probably want to start early. If you finish early, that's okay. But if you think about what we've covered during our discussions of chemistry, I think there's more material and it's also more complex than what we covered in chapter 1. Any other questions? Okay, then I will let you go for today. And as I say, we'll start out tomorrow. We'll look at the study guide, and then we're going to begin, kind of like we did last week, begin talking about what's on the test after this one. Our next big topic will be cells.